wheel. My research involves understanding how the brain actually is adapting to changes in sensory experience. So today I'm going to tell you one story about cross-modal synaptic plasticity, which is just a way the brain adapts to sensory loss. So as you probably know, that we experience our world with our five senses, and they are processed by very specific parts of our brain initially, which are called the primary, sensory cortices. And we process vision via the visual cortex and tactile information from the somatosensory cortex and so on. And these kind of very conventional textbook diagrams sort of mislead us to believing that these are very discrete areas that actually don't interact with one another. But there's a lot of evidence accumulating these days that they actually can interact with one another and are influenced by the other senses. And in particular, I'm interested in understanding how does the brain adapt to losing one of the sensory modalities. So when we're actually experiencing the world, we are living in a multisensory environment. So even a simple thing like watching a bird singing, you're basically doing a very complicated computation. So vision is captured by your retina, the sound is captured by your ear, and at some point down the process, you have to put them together to merge all of this sensory information to make the coherent view of the external world. So obviously, I'm not interested in understanding how that is happening, but what I am interested in understanding is what happens when you lose one of these sensory modalities. That is what happens when you lose vision. How does the brain adapt to not having a sensory input? And there's a lot of anecdotal and experimental evidence suggesting that there's something going on centrally that allows the individual to process the remaining senses better. This is called cross-modal plasticity, and that's my central topic for today's talk. So we now know the cross-modal plasticity can happen even in the very primary sensory cortical areas, like the primary visual, primary auditory, and primary somatosensory cortex, which are thought to be very early stages of sensory processing. And in particular, we now know there's two distinct types of adaptation, one that happens in the deprived cortex, such as the visual cortex, when you lose vision. We call this cross-modal recruitment, and I'll explain to you why that is. And also there's a different set of plasticity that's engaged in the remaining sensory cortical areas like the auditory or the somatosensory area. And those are called compensatory plasticity. And we'll talk more about the differences between these type of plasticity and how they can allow the brain to process the remaining senses better. So one of the critical things about this cross-modal recruitment was this very intriguing finding early on in the mid-1990s when people started doing functional brain imaging in humans. So this is one of those such experiments where they were using PET scan to look at activity in human brain. So this is cited in the visual reading print, and this is the area of neural activation that's highlighted in red, which includes the primary visual cortex as well as other visual cortical areas. And no surprise, reading print is a visual activity. But this is what they saw when they imaged a blind individual reading Braille, which is actually a tactile input, as you may imagine, and you can see as much activation in the visual areas, including the primary visual cortex. So these kind of things sort of led to the idea that there's something going on in this deprived cortex that is now getting recruited to process the remaining senses, such as tactile information in this case. Initial studies suggested that this might happen in early onset vision loss or blind people, but it turns out you can get this kind of adaptation pretty rapidly, even in adults. So this is a more recent study where they've been blindfolding adult individuals while they are trained to read Braille, and they were doing functional MRI imaging of brain activity while they're learning to read Braille. And you can see after about 5 days of learning how to read Braille, blindfolded, you can start getting activation in the primary visual area. And when the blindfold comes off, then basically you lose that activity. And in this paper, they went on to test that these blindfolded people actually learn how to read Braille much faster than the visually sighted controls, but also that they did transmagnetic cranial stimulation, TMS, to disrupt this activity in the visual area to show that that enhancement in terms of learning how to read Braille is actually due to the visual cortex activity. So it turns out the adult brain can undergo these kind of changes in a rather rapid fashion, which was quite unexpected. Another thing why I'm interested in studying this kind of mechanism is because of the complications we have when we're trying to recover a lost sense. So I'm sure a lot of you are familiar with this artificial cochlear implants, which is done to recover hearing in deaf individuals. And when these kind of surgeries are done pretty early on in life, the individual can regain speech perception quite readily. Even though these are very crude electrodes, normally I think they have 8 context points. I think now it's up to 16 or 64, but still it's a very degraded signal, yet they're able to use that to decode speech. But when these kind of surgeries are done later in life, then the speech recovery isn't great. So that's been attributed to the fact that the auditory cortex is now being recruited to process these other senses by this kind of cross-modal recruitment. So we think by understanding these kind of mechanisms, we might be able to enhance it to, accelerate the adaptation to losing a sensory modality, or, reverse the changes so we can recover function when we're trying to use these kind of neural prosthetic devices. So the model system we use is a rodent. Usually we go with mice because of the available genetic tools, and they have their sensory cortices arranged very similarly to humans. So they have the visual cortex in the back of the brain, and then auditory cortex here, and some of the sensory cortex located as diagrammed here. And we deprive vision by simply putting the animals in the dark for a few days. And the advantage of blinding them this way is that we're not damaging any of the optics, such that we can bring them back to light to see if the recovery process is there. And of course, in this particular case, when we're depriving vision, we'll be looking at plasticity that could be cellular basis for cross-modal recruitment in the visual cortex. And we looked at A1, which is the primary auditory cortex, to look for signs of compensatory plasticity. So to study this, we have to do recordings of synaptic transmission. These are all glutamatergic synapses that we're looking at. So they involved AMPA receptors and NMDARS receptors as other synapses. And the canonical feed-forward circuit that leads the information into the sensory cortices is very similar across sensory modalities, except olfaction, where they go through the thalamus, which then arrives to the cortex in the layer 4 initially, and then they move to layer 2, 3. And these feed-forward synapses are the synapses we're initially interested in studying, but then it turns out, even though the main function of the cortex is to process information coming from the periphery, the number of synapse-wise, these are actually a minority. They're estimated to be about only 5 to 20% of total number of synapses onto these neurons. And the majority of synapses are from other sources, intracortical or even from subcortical sources. So in order to study these very sparse inputs, we utilize optogenetics to target very specific inputs to determine whether there's changes in the synaptic transmission or strength of synapses after depriving vision. So the first layer that we wanted to look at was the thalamocortical inputs into these cortical areas. So we did the viral transfection mediated expression of channelrhodopsin in the visual thalamus and the auditory thalamus. And after several weeks of incubation to express enough channelrhodopsin in the terminal, we would make cortical slices and do acute wholesale patch clamp recordings to do measurements of synaptic strength. So once we do the experiments in the normal visual sighted animals, as well as the ones that were put in the dark for 1 week as adults. And this is to just demonstrate that channelrhodopsin works really well. And this is the layer 4 visual cortical neuron we reported from, that's filled with red, that's receiving all the channelrhodopsin expressing terminals that are shown in green. Similar thing with the auditory cortical recordings as shown here. And what we found was that there are changes in synaptic transmission. And I just want to briefly mention for people who are doing electrophysiology that we have to use a very interesting trick to desynchronize evoked release such that we can actually quantify single vesicle events. And this is actually necessary because we're using a viral transfection method. So the expression level of channelrhodopsin varies across animals as well as slices. And also the density of the axons that we can actually activate would be different across even different slices from the same animal. So we wanted to make sure we can quantify and compare the strength of synaptic transmission without the complications of viral expression. And the only way we thought was meaningful was going to be comparing a single vesicle event, because no matter how many terminals you're activating, you're only going to see a single vesicle mediated synaptic transmission. So we use this trick to desynchronize and release. So we're quantifying these little minis, as we call it, which are single vesicle mediated synaptic transmission. So just to give you an intuitive feel as to what this actually is that, whoops, sorry, I'm going too fast, is that it's basically we're measuring strength of an individual bouton releasing a single vesicle onto a postsynaptic cell, okay? So that's the synaptic strength we measure. And this is the result from our initial study looking at the thalamocortical inputs. We did not see any significant change in the strength of the inputs from the visual thalamus to the primary visual cortex, but we saw an increase in the amplitude. You can see it here, as well as the average data, that there's a strengthening of connections from the auditory thalamus into the primary auditory cortex. And that's reversed by a week of light exposure. So this was quite exciting as well as perplexing for us, because it's been known that the thalamocortical synapses stop having plasticity after the initial postnatal development, which we call the critical period. So seeing this change when we deprive the animals of vision as adults was quite a surprise. And moreover, the fact that we saw change in the auditory cortex, but really not much change in the visual cortex, was another surprise. That sort of meant that somehow the adult auditory cortex is paying more attention to what's happening to vision than audition. And the visual cortex doesn't care if it doesn't have any vision. But in either case, this was a very interesting finding. So then we decided to use a cell type specific expression system using the Cree transgenic lines to target different synapses. So for instance, we used a Cree layer 4 Cree animal to express channelrhodopsin just in layer 4 neurons. And this shows you one of those slices. We can see nice expression just in layer 4. And we did some quantification. And I guess I'm just going to skip over to the summary graph, because I took out most of the data because I don't want to bore everybody. But we used the same method using strontium desynchronized single vesicle events to map all of the different synapses in the cortex using this Cree dependent cell type specific expression. So what's shown in pink are the synapses that potentiate after 1 week of visual deprivation. What's shown in blue are the ones that actually depress after a week of deprivation. And what's shown in yellow are the ones that did not change the synaptic strength following visual deprivation. What's shown here are the results compiled from our recordings in the primary visual cortex. And these are results from the primary auditory cortex. So you can sort of see that there are lots of different colors everywhere, and you might think, oh, it looks just like a Christmas tree. So what's the importance here? But there is some pattern here. So I'm going to go over the basic pattern. So if you look at the visual cortex, the main change is strengthening of intracortical inputs. There's really no change in the feed-forward circuit, as you can see here. There's no change from the thalamus or layer 4 to 2-3. There's no change in local inhibition. But these intracortical synapses are potentiating. On the other hand, in the spared cortex, the auditory cortex, we see the opposite. We actually see potentiation of the feed-forward circuit at the cost of intracortical synapses because they're getting depressed. So this means the cortex has two different modes of processing, at least from what we can tell. That is, it has two competing inputs coming from the sensory pathway up and intracortical synapses, which will be bringing in they're not their own sensory modality, right? So it'll be some contextual information, as we call it. So the cortex has to process these two under normal circumstances, but then if you're missing the bottom-up sensory input, now they're potentiating synapses coming from other sources. such that they can now use that information to process. So we think this might be a basis for cross-modal recruitment because many of these intracortical inputs will be multisensory in nature. Whereas in the spared cortex, they're actually reducing those multisensory influences, but they're promoting the processing of feedforward information from the thalamus up. And we think this might be important because they're gearing towards making the system much more sensitive to sensory information coming through that stream. But obviously there's something that's being lost such that we're not gaining everything. So in some sense, the brain is very, I guess they have to maintain some sort of balance. So you cannot gain everything. You have to gain something at the loss of something. So if you're losing your feed-forward input, you're going to gain the intracortical processing and vice versa. So in either case, we thought this would be, interesting to, further characterize, because what does it mean that you're actually potentiating the feed-forward circuit? And just a straight naive, thought was, since you're potentiating the synapses carrying the auditory information, our prediction was the threshold or neural threshold for sound in the auditory cortex will decrease. So we decided to do in vivo recordings to see what is the tuning properties and response properties of these auditory cortex neurons. So we collaborated with Patrick Canonical, who's now here in the BME department at Hopkins. And he basically did single unit recordings from auditory cortex layer 4 neurons. The basic finding is summarized in this complicated figure from a paper, but I'm just going to go through very simply. So what we did was, these are basically tuning properties of individual auditory cortical neurons in the normal animals as shown up here. Dark exposed animal example is shown here. So the threshold for detecting sound is actually lowered in the dark exposed animal. The tuning curve itself is narrower in the dark exposed animal. And these are the group data shown on the bottom panels. So simply put, the neurons can respond to softer sound because their threshold is lowered, and they should be able to discriminate different tones better because their tuning curves are much sharper. So that sort of supported the idea that this type of plasticity might be able to allow the auditory cortex to become more sensitive and much more refined in terms of what they process in that stream. Whereas on the other hand, the changes we see in the visual cortex, we think might be responsible for this cross-modal recruitment. So we're currently figuring out what are the sources of these intracortical inputs, because we expect them to come from other sensory areas or multisensory areas. So that's one current experiment that we're doing right now. So in addition to all of this circuit mapping, of course, I'm a plasticity person, so I'm trying to understand how are these synapses changing. So we decided to look at some of the cellular mechanisms underlying this plasticity. So the first thing we looked at was what's happening to these intracortical synapses. And we hypothesized this might be a homeostatic adaptation to not having vision. And the rationale there was, since they're not getting any visually driven activity, the visual cortex will become more silent than previously. And by becoming inactive or less active, we hypothesize that they might be calling in some homeostatic regulation to potentiate the synapses that are still receiving input. So I'm going to tell you two different models for homeostatic plasticity that we initially thought might be responsible for this kind of adaptation. And one of them is called the sliding threshold metaplasticity model, and the other is called the synaptic scaling model. And this is coming from a recent review that I wrote with Alfredo Kirkwood, where we proposed that the sliding threshold model could be operating within a physiological range. And what sliding threshold model is that basically the strength of synaptic modification, which is plotted on this y-axis. So negative portion will be synaptic depression, long-term depression. Positive portion will be long-term potentiation, LTP. And basically what the function shows is that initially there's going to be a level of activity that's going to trigger LTD. At some point, it crosses a threshold where the activity would now trigger LTP induction. And the important thing about this sliding threshold model is that this threshold where the crossover occurs between LTD and LTP, it by itself is modified by past activity. So if the neurons have been silent for a while, in the original model that was proposed by Leon Cooper's group suggested hours to days, then this threshold will slide down when there's less activity, such that the LTD range shrinks and you can induce more LTP to compensate for lack of activity, because now some of the synapses will cross this lower threshold to become stronger so they can drive the neurons to recover their activity. When the neural activity is high for a long time, theta M will slide up such that the LTD range increases at the expense of LTP. So most of the synapses will get weakened such that it will renormalize the average activity of the neuron. So it's a very powerful model that was proposed by Leon Cooper's group in the 1980s. On the other hand, when the neural activity is really high, such that it cannot operate with the sliding threshold, I'll go why that could be, then we propose that synaptic scaling model, which is a global adaptation of synaptic strength, would be kicking in to bring back the homeostasis. So let me go and explain those models side by side. So the sliding threshold model, as I briefly explained, involves this threshold of synaptic modification moving back and forth as the function of past neural activity. So we posited that if there is a loss in major input, as would happen in the visual cortex when you lose vision, the theta M will slide down such that you're going to be promoting LTP and then removing some portion of LTD. So previously, if there were two inputs that were in the LTD portion, one of them, which is stronger, would cross the new threshold to become potentiated, but the other one will be kept low such that it will still weaken by LTD mechanisms. On the other hand, synaptic scaling model posits that all of the synapses will be globally scaled up and down by the same multiplicative factor. So let's say A1 through AX are all of these strength of individual synapses. Some of them will be larger than others because they have undergone LTP. Once you scale them using these multiplicative scaling mechanisms, the relative difference in strength of these synapses are going to be kept the same. So let's say A1 is twice stronger than A2. That will still be kept the same ratio here, just that all of them will be either stronger or weaker by the same factor. So you can sort of imagine that in this case, the plasticity will be input specific because now input 2, which used to be weaker, is going to be stronger, and this will still be weak. Whereas in this case, if A1 and A2, one was stronger and one was weaker, they will both get stronger after the change. So there's input-specific adaptation, whereas this will not be input-specific. The other major difference is that this still acts on LTD and LTP, which is NMDA receptor dependent. So the outcome of the synaptic change would still depend on NMDA receptors, whereas in the synaptic scaling model, these changes have been actually demonstrated to occur independently of NMDA receptors. So we decided to use these two key properties to test whether the changes we see with the cross-modal adaptation is sliding threshold or synaptic scaling. So first we tested the input specificity. So the visual cortical neurons we're recording from, which is layer 2-3, received two distinct inputs. One is a feed-forward input coming from layer 4, and then intracortical inputs coming from other sources. So we decided to use optogenetics to activate layer 4 neurons using the layer 4 Cree to drive channelrhodopsin. And then we place an electrical stimulation laterally to activate these intracortical inputs as a bulk. And then again, we're using the light-evoked desynchronized minis to measure the synaptic strength coming from either of these inputs. These are the results from intracortical synapses. So when we electrically stimulate them, you can see that visual deprivation, dark exposure, makes these synapses stronger, and then light exposure brings them back to normal. In the feed-forward pathway, on the other hand, we did not see that modification, meaning that this is very selective for intracortical synapses, but there is no change in the feed-forward circuit. So the change is input specific. So next, we tested the NMDARS dependence of this to distinguish the two models. So we used a genetic tool to knock out NMDA receptors acutely and very specifically in layer 2-3 neurons by sparsely transfecting Cre-GFP into the visual cortical layer 2. And this is just an example recording we did from one of those slices where we had targeted one of the cells expressing the Cre because it's labeled with GFP, and another cell that did not have the Cre-GFP. And we verify that the one that is expressing GFP are lacking NMDA receptor current as shown in this trace and quantified here. Neighboring cell, which is this guy, still has NMDA receptors just like the control cells. So this is a very acute way to knock it out because we knock it out. So the Cre is injected about 2 to 3 days before the experiment. So we don't have the long-term consequences of not having NMDA receptors. So in either case, when we looked at the control cells, which are expressing GFP construct alone versus the one that we have the Cre-GFP to knock out NMDA receptors, we see the following. So these guys have normal increase in synaptic strength with visual deprivation that is reversed, whereas in the NMDA receptor knockout cells, we don't see that regulation. So it is NMDA receptor dependent. So again, we now think that this is the model that supports the changes in the visual cortex based on these experimental data. So we don't think it's happening via a synaptic scaling mechanism. Let me just check the time because I want to make sure I'm not going too long because I did prepare an hour talk, but I realize I have to give 30-minute talk. So let me just briefly explain this and then I will try to wrap it up as I can. So how does the sliding threshold model mediate cross-modal plasticity is what I want to leave behind here. That is, we think it can explain both modes of changes. In the visual cortex, you can imagine when you lose vision, the major input disappears, meaning the threshold for synaptic modification will slide down, meaning the second best input, which used to fall below the threshold to be weak, now can cross the threshold and become stronger. And we believe this is what we're seeing in the intracortical inputs. So before it was weak, but then after vision loss, these guys are potentiated such that they can now bring in multisensory information to B1. In A1, we think the opposite is happening. So, normally their thresholds stay around here, but for some reason, we think the threshold goes up. I'll tell you why I think that way, because the feedforward circuit is getting stronger. So basically the thalamocortical inputs are potentiating, layer 4 to 2-3 inputs are potentiating. So there's more activity coming through the circuit. So the theta M would actually get bigger, meaning that before, if there are two different frequencies that can activate this neuron because they're both crossing the threshold, now one of them will get depressed because it's now falling below the new threshold. Therefore, you'll be only selective to one of them. So this can narrow the tuning curve on its own and also refine the feature in which the neuron would detect. So we believe this will be beneficial for this compensatory plasticity in auditory cortex because it will allow more refined processing of the auditory inputs. So this is basically what I've summarized previously. And one last thing I wanted to show you, but I think I'm going to just skim through the data because I don't have enough time, is to go to this part, which is what's happening here, because we've shown that there is potentiation of thalamocortical inputs. And I told you that this was a big surprise because we're seeing this in adults where this kind of plasticity shouldn't happen. And we have evidence that this actually is LTP, recovery of LTP. Of course, when you're trying to get LTP, the one thing that pops into your mind, hopefully, is that there has to be some kind of high frequency activity coming through a circuit or a lot of activity coming through a circuit. So then how is that happening when you don't have this vision? So initially, I thought naively, maybe there's more sound coming through a system, so there's more activity driving the circuit. And I thought maybe the way the animals, communicate by sound, they might be vocalizing more in the dark because they can't see anybody. So we did measure the sound level in the darkroom just to see if they were vocalizing more, but we didn't find any obvious difference. So we did a lot of analysis, but so far we cannot find any difference, at least on average. So we don't think it's the sound level, meaning that something is happening centrally to boost that sound such that there'll be more driving input to the auditory cortex. And I'm just going to briefly go over that. So one way to gate the sensory information through the thalamus is through this inhibitory nuclei called the thalamic reticular nucleus. So we have some data that just got published in Neuroscience. So if you're interested, you can go and see the data, but I'm going to skip through all of this to show you just the summary data. Because in the interest of time, but these are all the data that's in the paper, so you're not missing anything. So now this is the circuit I showed you now, and our data basically suggests this model. So when we looked at the TRN inhibition onto the visual thalamus, we did not see any change with visual deprivation. But then we saw depression of inhibition onto the auditory thalamus. And this happened by two different mechanisms. One is postsynaptic weakening of the inhibitory synapse in the MGB neurons, but also we found evidence that there is presynaptic depression, which is called ILTD, that is likely mediated by CD1 receptors on the terminals of TRN neurons, and they actually depress the synapse. So, of course, when you're depressing inhibition, that means more activity can go through the auditory thalamus, and I think that is driving this LTP in the auditory cortex. So one of the things that was kind of surprising to us, which I want to point out, is that the TRN is one anatomical structure, as we all thought, but it seems like they have different regulation depending on the target. So that the auditory thalamus is less inhibited by TRN when you don't have vision, but the visual thalamus, there's no change in this inhibitory strength. And this kind of specificity I wasn't expecting, because I thought it was kind of interesting how it could actually be doing it, but it makes a lot of sense considering that this is actually might be mediated by endocannabinoids. If you know anything about endocannabinoids, they're actually produced by activity from the postsynaptic neuron, and it's a retrograde messenger that goes to the presynaptic terminal to decrease the release. So we think that kind of specificity can be generated by the postsynaptic neuron. But then again, the question becomes, then why is the MGB neurons more active such that it can produce more endocannabinoids? The next question. So we need to probably go further down the chain and see the inputs into MGB and see if those are actually altered. So that's our next installment of the grant. So that's the one that I just got funded. In either case, as the last thing, I want to just show everybody who was involved in the work. So the initial work in the auditory cortex was done by my previous graduate student, Emily Petrus, who's now a faculty at Uniformed Services University down in DC. And the mechanisms, or the homeostatic mechanisms and the NMDARS receptor dependence was all done by my two former graduate students, Gabriela Rodriguez, who's a postdoc in Max Planck right now with David Fitzpatrick, and Varun Choksi, who's a postdoc in Dan O'Connor's lab here at Hopkins. And the TRN work, which I had barely had time to even explain what it is, was led by my previous postdoc, Jessica Witt, who's a teaching faculty up in Connecticut, with the help of my current graduate student, Gabrielle, and two very talented technicians who had worked in my lab and currently in my lab. And these are my collaborators here, and these are my funding sources. And thank you very much. And I'll be around to answer any questions.